Hi everyone, my name is Carson Gido and I am a junior at Kansas State University majoring in biochemistry. I work with Dr. Brian Geisbrecht in the Department of Biochemistry and Molecular Biophysics, and our lab studies the interactions between pathogenic bacteria and the human immune system. My research focuses on a class of molecules produced by Staphylococcus aureus bacteria called extracellular adherence proteins, which we abbreviate as EAPs. EAPs bind to and block the function of neutrophil elastase and cathepsin G, which are two proteases produced by human white blood cells that function to destroy invading bacteria. Prior to this study, we did not know the structural basis behind one EAP domain called EAP1's inhibition of elastase and cathepsin G. So our objective was to determine the structure of EAP1 bound to both proteases and understand why this inhibition happens. To do this, we used a method called X-ray crystallography. This involves isolating each EAP1 and protease complex, crystallizing it, shooting it with X-rays, and measuring how the X-rays diffract off the crystal. This information can be used to determine the structure of the protein complex with a number of computer programs. The result is the figure you see at the top right-hand corner of the poster. This shows EAP1 in blue and how it binds to neutrophil elastase in orange and cathepsin G in pink. The first interesting thing we noticed is that EAP1 uses two distinct loops on either side of itself to latch onto these proteases. These loops position themselves over the active site of the proteases, which is where most of the enzymatic work is done and blocks their function. If we take a closer look at the regions of EAP1 that interact with each protease, which are the figures at the bottom, we see that there are several interactions between the two proteins in either case, shown by gray dotted lines. These interactions are mostly hydrogen bonds, which act as non-covalent linkages that hold EAP1 and the proteases together. Finally, these findings have meaningful applications to the health of Kansans. Infections of Staphylococcus aureus, which produce EAP, are a major problem in Kansas hospitals, and excessive action of white blood cells, which produce neutrophil elastase and cathepsin G, have been shown to lead to COPD and lung cancer, which kill thousands of Kansans per year.